Good morning, Biology 102. Today we're talking about biotechnology. This is the manipulation of living things to make useful products. And specifically today we're going to be talking about DNA technology. So using and manipulating DNA in order to create useful products, typically for human consumption, right? Or for human use. Over here in my picture, I have a piece of corn with some pests on it. And one of the ways that DNA technology has helped us is in agriculture, where we've moved different DNA pieces into different seeds so that they gain things like pesticide resistance. So we'll be talking about that today. I want to begin by talking about something called restriction enzymes. This is a technology and an enzyme that recognizes a specific piece of DNA, so a specific sequence of DNA, and can cut DNA at that specific site. This kind of technology is going to mimic DNA proofreading or crossing over enzymes that cross over equal links, or proofreaders that go through and cut out mistakes and replace them with the correct sequences. So restriction enzymes allow us to cut out specific sequences of DNA. Over here in my slides, I have an example of a restriction enzyme that is cutting this length of DNA between the guanine and adenine. Oftentimes they're not quite this simple, but this shows you over here in this picture how that actually looks at the end of the day. Now, if we can cut out a gene of interest, like pesticide resistance, or maybe temperature resistant, like freezing resistance, things of this nature, and if we have vehicles to move that DNA from one location to the next, then we can create something called recombinant DNA. Recombinant DNA results when DNA from two different sources are combined into a single molecule. Often we're using things like viruses. Over here, my picture shows a virus injecting DNA, or sometimes a bacteria as our vehicle, or they're often called vectors, to move that DNA that we just cut out using restriction enzymes into a new source. This is at the heart of genetic engineering, manipulating genes and DNA for practical purposes, like for pesticide resistance in corn. Now, what can genetic engineering do for you? So what kinds of useful products can scientists make from this kind of technology? There are lots of different ways that we've used this historically. Over here in this picture, we're showing this black gene of interest here. We've taken it out of sort of the organism, and then we've recombined it into maybe like an E. coli bacterium. So you can see that we have this like new piece of DNA inside of this bacteria. We can either grow up this bacteria and harvest more copies of this gene, maybe pesticide resistance, that we then insert into plants or into seeds so that they show different traits for us. We can insert that gene into other bacteria so that we have different kinds of bacteria that can eat oil slicks or take care of pollution events, which we have mentioned before. We can also grow up this bacteria, and rather than harvesting the gene, we can actually harvest the protein product. And this protein product can then be isolated and sort of solidified, and it gives us different things, like it gives us ways for making different proteins that maybe break down clots before they can clot around your heart and cause like a heart attack. So we can harvest that protein so that it makes it where we have like a medicine that we can then give to a human that is having a heart attack. Some other ways that we use this is in developing cancer drugs. A lot of our crops are going to use genetic manipulation and genetic engineering in order to create those seeds that have different resistances inside of them. Insulin was the first case of this, so I want to talk about that, but we also use this for various drugs and hormones beyond insulin. But insulin, I don't know if you know this, but back in the day, before we could create and have genetically modified sort of bacteria to create insulin for us, we used to go to the slaughterhouses and we would collect pig and cow pancreases and then harvest off the insulin from those slaughterhouse situations. This was great, but it turns out that cow and pig insulin is a little bit different than what humans make. And sometimes your immune system cells would recognize that there was a difference. And so for diabetics that were taking cow and pig insulin, sometimes they would have almost like a rejection to that insulin because it wasn't human insulin. So in 1978, we actually modified a bacteria in the US. We cut out the gene for insulin. We moved it into a bacteria. Now we grow these vats of bacteria up and we harvest this product of insulin. Which is a protein product that we can harvest from our E. coli, and then we can sell it as insulin. The first insulin on the market was Humulin. And what was nice about Humulin over sort of harvesting these pancreases from pigs and cows is that it was human insulin. So if you move the human gene for insulin into a new bacteria and then you have it create this insulin for you, the nice thing about this is that it's actually human insulin. So there's less rejection and things like that. For this particular drug, lots more people can take it and they have success taking it because it's the human form. Another way that we use genetic engineering is to develop vaccines. Vaccines are harmless variants or derivatives of a disease-causing microbe that we can then inject into humans so that we build memory cells, immune system cells that remember certain protein codes or certain pathogens for the future so that they can deal with them more quickly. An example of this is hepatitis B. We have developed that particular vaccine based on moving a gene for the protein coat on the capsid of this particular virus into or onto a non-pathogenic cell. That cell displays that sort of viral coat so that our memory cells can then remember what that like sort of outside coat looks like. If they see it in your bloodstream and they've sort of developed these memory cells, then they can attack it more quickly and in hopes that you won't get sick, right? You won't get fevers or other sort of digestive issues or diarrhea, vomiting, etc. that come along with these kinds of diseases. So vaccines are often made this way, and the bulk of the vaccines that you have probably taken in your lifetime have been made this way. Another way we use genetic engineering is to modify foods and products in agriculture. This again usually constitutes cutting out, using restriction enzymes to cut out genes of interest like our pesticide corn gene, and then getting a corn seed to take up that gene and express some kind of trait. And recombining two sources of DNA into one sort of organism. I think in the US right now, about half the corn over here and 3/4 of the soybeans, cotton are genetically modified seeds that we are using. A lot of my farming students in the past have said that this enables them, this kind of genetic modification enables them to grow more seed in sort of smaller plots, get higher yields for their crops each year. And as our growing human population goes forward, having more crops available to humans is something to consider. There are other people who have different opposing ideas, and we'll talk about these a little bit more in our Zoom meetings sort of next week. But I also wanted to bring up some other reasons why we might use genetic modification. This is golden rice, and then over here is white rice. So this yellowy rice is a golden rice. And rice companies actually moved a gene that protects you from childhood blindness due to lack of vitamins into rice, so that it would express this like kind of necessary vitamins for developing your eye correctly. This was then given to different areas of the world where this vitamin is in lower amounts, right? So you don't find it as much in like sort of the soils and in the crops and whatnot. So they would have a higher amount of this sort of vitamin, so less in hopes of like kind of correcting for that childhood blindness that was a problem in those areas. So another reason why you might use genetic engineering. All right, so if we go to our slides, the next thing that I want to talk about is CRISPR-Cas9. CRISPR-Cas9 has been in the news for the last year or so, and there's a few human trials started in the US, and so you'll probably be hearing more about this as we go forward. But this is a gene editing system. CRISPR-Cas9 has a couple of different parts to it. It typically has a sequence that will find or guide our RNA piece or our system here to a specific location in a cell. And then it also has some nuclease type cutters, right, that can cut out a particular sequence of DNA. And then sometimes linked on the side of this will also be the correct sequence. So what CRISPR allows us to do is find different mutations in the genome, cut out the mutation which is problematic, and replace it with the correct sequence. The interesting thing about CRISPR-Cas9 is that it can be done in living cells. And so they're doing some fetal trials in some other places in the world, but they're also doing some cancer trials and some sickle cell anemia trials on adults and living cells in the US. And they have had some success. Some of the early results after a few months have shown that this particular editing system can edit and correct for a mutation. So the long-term studies haven't been done yet, but this is on the horizon. CRISPR-Cas9, I mentioned before, comes from bacterial and archaean cells. It's almost like an immune system defense against viruses that attack them. And so what they'll do, these bacteria and archaeans will actually chop up the viruses, and then if they get attacked again by those viruses, they know to sort of chop up those pieces. So this is almost like a beginning immune system from these viral attacks that they get. And we've hijacked that machinery, but changed the guide so that it guides to whatever sequences we're interested in, and then also gives replacement sequences where we cut out whatever we're interested in correcting, and then we can manipulate and splice in whatever sort of new code that we want to be expressed. There are also some systems being built that will help us detect pathogens. So there's some ways, those are called Sherlock Cas13, but these are also some ways where they're finding different mutations, things of this nature, and then detecting for pathogens in early stages, maybe before some other tests can detect those particular pathogens. So another way that we're using sort of gene editing and using these kinds of technology to sort of create some better technologies for humans. So DNA profiling, also known as DNA fingerprinting in some cases, allows us to analyze DNA samples to see if they're from the same person. This you might have heard of in forensic science and crime scenes, so the different shows on TV that show you how you can take small amounts of DNA from a crime scene and then compare it to your suspect's DNA to see if you get a match. A similar technology is also in existence for genetic testing. So if we know a particular mutation like sickle cell anemia or Huntington's disease, we can actually then develop a test so we can test people's blood or DNA for a specific genetic disorder. And lastly, we also use a similar kind of technology for developing our phylogenies. Remember those evolutionary trees where we have these groups of organisms and we would compare them? The DNA technology that we use that shows relatedness between those groups of organisms so that we can sort them and build our trees is a very similar sort of technique. So I want to walk you through some pieces or some things that we use in DNA profiling. So one of the things that we use is PCR. Polymerase chain reaction helps us copy a small amount of DNA into larger quantities. This allows us to take small amounts of DNA from a crime scene, like think of blood under the fingernails of a murder victim, right? And then copy it in a test tube over and over again so that we can run different genetic tests. So PCR just allows us to copy DNA and make millions of copies of it, right, very quickly so that we can run some tests. And one of the main tests that we do for these genetic tests, for phylogenies, for DNA profiling and fingerprinting, is gel electrophoresis. And in gel electrophoresis, we build a gel, and it almost looks like a Jell-O jiggler. I'll be filming something that you would have done in lab using these processes. But this gel then allows us to sort DNA based on charge and size across the gel plane. And this can allow us to tell the difference between suspects or relatedness within groups of organisms, etc. So DNA profiling, here's an example of a crime scene. We can, if this is our gel down here in this box, we can load in our crime scene blood, and we can load it in this starting well and then allow it to migrate over time based on size and charge. We typically, in our samples, will also put restriction enzymes, and restriction enzymes will allow us to cut DNA in various places. And because there are different sections of DNA that are variable for each individual on planet Earth, then this allows us to sort our suspects. So we can use a certain batch of restriction enzymes that cut DNA in a certain sequence, and then we can put those same restriction enzymes with all of our suspects' blood and then allow them to go on the same gel. The idea is identical, like sort of sorting positions means that the blood is identical or from the same individual. So if we look over here, lane number 1 with our crime scene blood, it doesn't quite match suspect 1. So suspect 1 has something unique about their blood, but suspect 2, all of these bands, we call them, are lines of DNA where our restriction enzymes have cut DNA into smaller pieces. Suspect 2 actually matches that crime scene blood. So it gives us a way to sort of sort and fingerprint suspects given what we find at a crime scene. When we develop genetic tests, this is showing sickle cell anemia. If we know the gene and the mutation mistake that is made in sickle cell, then we can develop a way to either cut if it's normal at that sort of position, or cut if that mutation is present. This is showing where we have a test where the restriction enzymes cut if you are normal. So they'll make an additional cut here if you are normal, carrying this hemoglobin sort of in development of the shape of your red blood cell. It will allow us to cut. And then if you have sickle cell sort of mutant B globin, then it doesn't make that additional cut. You can see that it has only 2 pieces if you have sickle cell, but if you're a normal person, then you would have 3 pieces. We can then load up this blood or with these restriction enzymes in our starting wells and run our gels. And then this banding pattern, again, allows us to see the difference. We can see that a normal person would make 3 bands along our gel, and a sickle cell allele, if you have that, would only make 2. So these genetic tests, as long as we know the sequence, we can develop some way to cut if that sequence is normal or cut if that sequence is mutated, and then we can develop a genetic test to give to people. The last thing that I want to mention, last technology, is cloning. Cloning we've already talked about. So we can cellularly clone using a process that is like mitosis, even in a test tube. So we can clone and copy DNA and PCR, we can clone cells, etc. But we're often talking about organismal cloning, so cloning a full organism when we're talking about the ethics relating to cloning. And so here I showed Dolly the sheep, 1996-97. Dolly the sheep sort of came to be down there and was one of the first sort of mammalian examples of organismal cloning. So how do we do this kind of cloning? Well, plants actually do cloning naturally. They often will have a regeneration or a regrowth of lost parts process. You know this because oftentimes if you've picked carrots or potatoes, you know that you can grow a full new plant from the eyes, right? The potato eyes or the carrot eyes. And so this is a way that agriculture can use this process where we take a group of cells and then we clone sort of plant lines from them. This can be good, right? There can be some positives from this. We can increase our yields. We know what we're getting in our crop plants. Orchids, I have up here, almost all of those that you buy in the store are clones. And what's nice about that is it takes the pressure off the natural populations, right? So that we're not hunting things to extinction in nature. So plant cloning, most people are like, yay, ethically, this is fine with them. And you have no like sort of qualms. Again, we'll be talking about some of the ethics in our Zoom meetings. Animal cloning, on the other hand, most people are okay if it's worms or things that don't have a backbone. But once we get to vertebrates, right, things that are close to ourselves or we have sort of that backbone in common, then people start to be like ethically a little bit more concerned about it. How does cloning happen? So usually there's nuclear transplantation. So usually we have to remove a nucleus from an egg cell. Egg cells are primed to divide correctly and to create a new organism. So you can't use like a skin cell or something like a normal body cell. You have to use something primed to sort of create this new organism. So we'll remove the nucleus from this egg, and then we can transplant in a nucleus from a donor. Once that is transplanted in, we can grow this group of cells. It will start to divide using mitosis, etc., into a mass. Usually in a mammal like yourself, it would take 4 or 5 days down the fallopian tube before it implants. So once the cells have grown up, kind of in that transitional period, then once they're ready for implantation, then we can implant them into a surrogate of some sort. And then wait for the gestation, and out comes like sort of this clone. We can also take these cells and create stem cell lines, and we do this in order to run tests, to grow different like sort of compounds and things like that. Stem cells are very flexible cells, so they haven't been differentiated or determined. So sometimes these have uses for us in science, or we can go all the way to that full like sort of clone. 1996, we produced Dolly the sheep, and there was lots of controversy at the time. So again, some of the controversies of this surrounding organismal cloning have to do with, yes, whether we should be cloning these kinds of groups of organisms, livestock, etc. Also, should we be cloning humans, right? And what's the benefits or the negative sort of components of that particular, cloning event. We will be talking about more of the ethics, and I'll have some ethical questions for you to consider when we get to our Zoom meetings, but otherwise you can start thinking about them. So thanks for watching this video on biotechnology. I will see you again next time.